Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. Before we dive into today's fascinating discovery, please take a moment to like this video and subscribe to the Sand Science Experience. Your support truly helps us bring you more cutting-edge science and allows us to bring you these in-depth explorations. Today, we're embarking on a deep dive into something truly astounding happening at the microscopic level. It has huge implications for your cellular health. How two tiny powerhouses within your cells work together in a kind of secret cellular handshake to fight off stress. Now, you've almost certainly heard of mitochondria, right? The undisputed power plants of the cell. But what about their less famous, but, well, equally vital cousins, peroxisomes? It turns out they're forming this crucial dynamic partnership that completely redefines how we understand cellular defense.
Yeah, it's really fascinating. For a very long time, the prevailing wisdom in cell biology was that mitochondria were pretty much self-sufficient, especially when it came to cleaning up their own messes, those dangerous byproducts of energy production, reactive oxygen species, or ROS. But scientists have always had this nagging question. What if there's a vital, direct support system happening right under our noses, involving another organelle? Maybe something critical to keeping mitochondria healthy. It's been a bit of a puzzle.
And that's exactly our mission for you today, to unpack a groundbreaking new paper that reveals this precise cellular handshake, identifying the specific molecular players involved and showing us how this incredible collaboration keeps your cells healthy and resilient. Get ready for some genuine aha moments about the hidden world within us. So let's set the stage, shall we? We know mitochondria are essential. Everyone knows that. They're constantly generating the energy your body needs through a complex process called the electron transport chain, the ETC. But this energy production isn't always a perfectly clean operation, is it?
That's right. It's definitely not. You can think of it like a factory. It's incredibly efficient. But a natural byproduct of all that metabolic activity is the formation of reactive oxygen species, ROS. These are highly reactive molecules, like metabolic exhaust fumes, you could say. Now, in very small, controlled amounts, ROS can actually act as important signaling molecules. They help cells communicate and respond. But when their production overwhelms the cell's defenses, well, that's when you run into trouble.
And what kind of trouble are we talking about exactly?
Well, too much ROS leads to what we call oxidative stress. You can imagine it like rust forming on the vital machinery of your cell. It can damage proteins, lipids, even DNA. Ultimately, it impairs cellular function and can lead to cellular aging and disease. So maintaining redox homeostasis, that incredibly precise balance between these oxidants and the cell's antioxidants, is absolutely fundamental for cell health, survival, and preventing a whole host of problems.
Okay, so historically the view was mitochondria had their own internal cleanup crew for ROS, their own antioxidant systems. They handled it themselves, sort of the lone wolves of ROS defense. But you're saying there were hints this wasn't the whole story?
Precisely. For decades, actually, there's been this curious observation. Yeah. Almost a paradox, really. Scientists noticed that problems in peroxisomes, these other organelles, could somehow lead to ROS-mediated mitochondrial damage. It was puzzling. If mitochondria are supposed to be self-sufficient, why would peroxisome dysfunction mess them up? It suggested a deeper connection, something we didn't appreciate. This new paper really starts to unravel that mystery.
Okay, right, so let's unpack that then. Peroxisomes. What exactly are they and what's their usual job in the cell? They're clearly important even if they're not as famous as mitochondria.
Oh, they absolutely are important. Peroxisomes are ubiquitous single-membrane bound organelles. You find them in pretty much every eukaryotic cell, yeast, plants, humans. And what makes them incredibly special for this discussion is that they are absolutely packed with antioxidant enzymes. The most notable one is Catalase. It's uniquely efficient at breaking down hydrogen peroxide, which is a really potent ROS. They maintain this very reducing environment inside themselves, meaning they're basically primed to neutralize these damaging oxidants.
Yeah.
So given this powerful antioxidant capacity, the hypothesis started bubbling up. What if they weren't just general detoxifiers? What if they were directly involved in managing mitochondrial ROS, maybe acting as sinks for that mitochondrial exhaust?
A powerful cleanup crew, perfectly designed for the job. Okay, but here's the key question, right? If mitochondria make the ROS and peroxisomes are the cleanup crew, how do they actually get the ROS from the mitochondria? Is it just signaling or is there a physical connection? That feels like the core issue here.
That's exactly the critical question this study tackled. They looked into something called membrane contact sites.
And here's where it gets really interesting. Membrane contact sites. They're like physical bridges between organelles, right? Tiny connection points. We know they're important for moving things like lipids, metabolites, ions around quickly, super efficient communication. But could they also be a direct pipe for transferring ROS, like an antioxidant superhighway? That was the big unknown.
Exactly. And this study went right after that question. To do it, they developed this really clever spatiotemporal imaging method. Think of it like a tiny cellular GPS, watching these organelles move and interact in living cells in real time. They used some pretty advanced microscopy to measure the incredibly close proximity, the apposition, and the actual physical contact between peroxisomes and mitochondria. They could track the distances over time.
So they could see them actually touching, not just bumping into each other randomly.
Precisely. They were very careful to distinguish between just fleeting encounters and real contact events. They defined contact as a stable closeness lasting longer than about 31 seconds. A time frame, they figured out, meant a real interaction was happening. And what they found was remarkable. A direct physical link between peroxisomes and mitochondria. And crucially, they figured out which specific molecules make this connection.
That's the real breakthrough, isn't it? Naming the handshake partners.
It's exactly right. They identified two key proteins acting as the tethers. There's ACVD5, that's a peroxisomal membrane protein, and PTP-IP51, which sits on the outer mitochondrial membrane. These two proteins directly interact, mainly through these specialized structures called coiled domains. You can kind of think of those like molecular Velcro or zippers. They allow proteins to specifically grab onto each other and form these stable connections. So they basically zip together, creating and regulating this peroxisome-mitochondria contact site, the PO-Mito contact. And they proved it too. If you use techniques like SiRNA to get rid of these proteins, the contacts just dropped off significantly. If you made the cell produce more of them, you got more contacts. It's specific, it's protein-mediated, and it's regulated.
Wow. And what's truly fascinating here, even beyond just knowing this contact exists, is that it's not static. It's not always there at the same level. This cellular handshake actually responds when the cell is under stress, right? It becomes more common.
Oh, yes, and this is such a critical piece of the puzzle. It shows how dynamic and adaptive the system is. The researchers saw that the percentage of these PO-Mito contacts went up significantly when cells were dealing with mitochondrial oxidative stress. And they induced this stress in different ways, not just artificially with chemicals like Rotenone, but also using more physiologically relevant methods, like growing cells in galactose or palmitic acid, which naturally makes mitochondria produce more ROS. This dynamic regulation strongly suggests the cell actively increases this contact when it senses danger, like calling for backup when the factory fumes get too thick.
Okay, so if this contact ramps up under stress and seems so important, what happens if it's missing? What's the fallout if you, say, knock out that ACVD5 protein one half of the Velcro?
That's the vital question, isn't it? And they looked at that directly. They studied cells where ACVD5 was genetically deleted, these ACVD5-KO cells. And without that critical PO-Mito contact, well, the cells show this generally elevated oxidative state throughout the entire cell, much higher stress levels overall. For instance, they measured more oxidized glutathione in the cytoplasm. That's a key sign. The cell's main antioxidant defenses are being overwhelmed.
Yeah.
They also saw increased nuclear NRF2. NRF2 is like the cell's master alarm switch for oxidative stress. It turns on a whole bunch of protective genes. So the alarm was clearly going off.
So the whole cell is basically screaming stress when that connection is broken.
Exactly. And the mitochondria themselves really suffered. They became elongated, which is often a sign of stress and dysfunction. Their membrane potential dropped, meaning they weren't producing energy as efficiently. And crucially, they had higher levels of oxidized glutathione and hydrogen peroxide inside the mitochondria, especially when challenged with even more stress. All these problems together strongly suggest that this PO-Mito contact is protective. It's a crucial defense mechanism for keeping mitochondria healthy.
Okay, so the evidence is really piling up. The contact forms, it increases during stress, and without it, the mitochondria suffer badly. This points very strongly to a direct transfer of ROS, right, from the mitochondria straight to the peroxisomes.
Absolutely.
Yeah.
The data overwhelmingly supports that. This PO-Mito contact directly facilitates the transfer of ROS produced by mitochondria straight into peroxisomes. They even used a special reporter molecule to measure oxidation inside the peroxisomes. And they found peroxisomal oxidation went up when mitochondria were stressed. But here's the kicker. That increase was entirely dependent on the ACVD5-mediated contact.
Right.
If you disrupted the contact, the ROS transfer to peroxisomes was severely impaired. So peroxisomes aren't just mopping up random ROS floating around. They are specifically pulling ROS directly from stressed mitochondria through these intimate contact sites. It's a targeted cleanup.
A targeted cleanup crew. So peroxisomes are acting as these reducing sinks for mitochondrial ROS, pulling the toxic stuff away directly. And there's a real elegance to this system, isn't there? Something about how peroxisomes do the cleanup being very efficient for the cell.
That's a really great point, and it highlights the cell's incredible economy. See, unlike the glutathione system, another major antioxidant pathway which uses up NADPH to regenerate itself, the breakdown of hydrogen peroxide by catalase and peroxisomes does not consume NADPH, and NADPH is like prime cellular currency. It's a vital reducing agent needed for tons of other essential processes, like making lipids or detoxifying drugs. So by not using NADPH, the peroxisome pathway is this incredibly efficient, less resource-intensive way for the cell to handle antioxidant defense. It saves that crucial NADPH for other jobs. It's a really smart, economical solution.
So let's bring it all together. What does this really mean? This deep dive has shown us this amazing, previously unknown layer of how mitochondria defend themselves. It's not just a solo mission, it's a finely tuned collaboration, a multi-organelle process where peroxisomes literally reach out, form a connection, and pull damaging ROS away from mitochondria when they're under stress. A cleanup crew on demand.
Exactly. This discovery really transforms our basic understanding of how cells handle oxidative stress. And the implications are pretty significant, especially when we think about human diseases. Think about peroxisomal disorders. Patients with these conditions often have widespread mitochondrial problems. This study offers a compelling reason why. The issue might not be the mitochondria themselves failing first, but the breakdown of this vital communication, this ROS transfer pathway mediated by the contact sites. It totally shifts how we might think about treating these diseases.
It really does. And it makes you wonder, doesn't it, if these specific cellular handshakes are so crucial for managing internal stress? Well, are there other hidden connections? Other ROS trafficking pathways between organelles that we haven't even discovered yet, all working behind the scenes to keep us healthy? Definitely something for you to ponder as you go about your day. We hope you enjoyed this deep dive into the microscopic world of cellular defense here on the Sound Science Experience. Don't forget to like this video and subscribe to the Sound Science Experience for more cutting-edge research breakdowns and scientific explorations. We genuinely appreciate your support. Look forward to our next deep dive.